Institute. She did her bachelor's at the University of San Diego and her master's and PhD at the University of Hawaii at Manoa, and is now at Woods Hole, where she studies a lot of different aspects of coral microbiology and also looks at the respiratory tract bacteria in whales, which is a cool project she's not talking about today. So I'm really happy to have her here, and I hope a lot of you are going to be able to make it to her workshop later on, which will be really fun. So, yeah, thank you.
Thank you so much, Allison, and thank you to Monica as well as the Microbiome Center for hosting my visit. This is my first time in Penn State, which I'm actually really excited about. I love all the greenery around here, and everybody's been really hospitable, so thank you. Let's see if we can get my title slide up. Did it just go away? There we go. Okay. So what I'm going to be talking about today is the secret life of coral microbiomes. Yes, I spent way too much time on this title slide. For full disclosure. Has anybody seen the movie, The Secret Life of Pets? Yes. Okay, a few of you out there. So what I'm going to be telling you today kind of resembles some aspects of the plot of that movie. So if you haven't seen the movie, it's an animated cartoon about some pets, some dogs and cats that have the life that the humans observe, which is normal to what you and I see with our pets. And then they also have the secret life, a total underground world where they're involved in all kinds of activities. And so that's the story that I'm going to tell you about today with coral microbiomes, that there are a lot of secret lives going on. This is a major thrust of research in our lab, and so I'm going to tell you about 4 main secret lives that we've uncovered in our research. So to get started, hopefully this will work. Awesome. I'm going to show you a movie. I'm going to take you underwater, sorry, not underground, to a coral reef. There are no tropical coral reefs in Woods Hole, Massachusetts, so we typically find our way to nice tropical locations such as Micronesia and often work on boats like this, if we're lucky. This is the Aleutian, which is fully set up for scuba diving operations. So I was lucky enough to go out on this vessel to Micronesia a number of years ago, and we also had a wonderful film crew with us to document the reefs. So this is a really nice opportunity for me to introduce coral reefs to those of you who are not familiar with this system. So coral reefs are the tropical rainforests of the sea. They're home to over 9 million different species, and corals form the framework for this ecosystem, for this environment. Corals are indeed animals. They are animals composed of thousands to millions of different polyps on a single colony, and they secrete calcium carbonate skeletons. And so that is the material that's forming this reef. What you're mostly seeing, obviously, in this reef are the corals. There's not a lot of fish, quite honestly, in this environment. But what we can't see is quite a bit of other life that's on the reef. So within the crevices of the reef are nooks and crannies where you have many invertebrates that reside, especially snapping shrimp, which you might hear if you're underwater on this reef and not listening to the music going on. The other part of the unseen world of a coral reef is the microorganisms. So we know that there's microorganisms associated with corals. There's also microorganisms residing in that water overlaying the coral reef. Now, one of the reasons that I'm so interested in coral reefs and their microbiomes is because if we were to take the city of Manhattan and all of its inhabitants and all of its activities and put it in the desert where there's basically no other resources, that's basically what a coral reef is. It's this ecosystem in the desert that's surrounded by very little nutrients in the seawater. It doesn't have a lot of resources. It doesn't have a lot of food for these organisms to survive on. So these corals and the organisms on a reef have gotten really good at utilizing their resources, at recycling, and working together at forming cooperations. And microorganisms are central to those activities, and we're just really at the beginning of trying to understand what their role is on these reefs. So now that we're back on land, microbiology is certainly not new to coral reef science. There's been many years of research focused about 15 years ago and prior early 70s to about even going on now, focused on disease. And so this is part of my secret life story. And that if you were to go down on a reef and think about microbes, most of you wouldn't even think about microbes until you saw something like this happening to a coral. So these are corals with obvious bands of infection, of disease lesions, which have some microbial role. Some of this is still not known as to what's actually causing this, but these are the obvious signs of microbial interactions on reefs. The last 15 years we've had a shift towards understanding the microbes associated with these more healthy-looking corals. So on these reefs, you don't see any obvious kinds of microbial infections. You don't see signs that something might be amiss. And so most people don't really think about microbes playing a role in this ecosystem. And it was in early 2000s, 2001 and 2002, when Forrest Grower, a scientist in San Diego, published what I think are two really seminal papers that describe the coral microbiome, the bacteria that were associated with coral, the archaea that were associated with corals, and found that they were extremely diverse. Each coral colony home to 1,000 to 6,000 different species of coral, each colony having its own unique signature, but links between colonies in different oceans, between species associated with colonies in different oceans. So there seem to be some themes emerging here that this coral microbiome was potentially very abundant. It was potentially linked to something that the coral itself needed. And so that is what spurred my interest into coral microbiology, at least looking at the prokaryotic side of the association. And I'm glad I went into this research area because when there's so much unknown, there's just so much to know, right? So hopefully this has worked out okay for the time being, and I'm hoping that it will still continue to be a fruitful career. So the microbes that I'm going to be talking about today are just a small component of the total coral microbiome. Let's see if we can advance. The microbiome component that has been well studied for many years, even before the bacteria, are the algae. These are the dinoflagellates that form endosymbiotic association with corals. These are highly successful symbioses. The algae is responsible for transferring as much as 90% of their photosynthetic product to the coral, and many of these corals do not survive without this association. There's so much to know about this association, and it's an extremely important association, especially as corals and their algae are disassociating in a phenomenon known as bleaching. We've seen a lot of that recently. And so there's quite a bit of research going on in this area, which is really important for the longevity of this association. What we know less about are the associations that corals have with prokaryotes, bacteria, and archaea. We know that these cells are very successful in other environments as harnessing nutrients that are available in the environment, especially things that are present in low concentrations. We also know that bacteria can produce and secrete antibiotic-type compounds, which help prevent pathogenic infection in these types of environments. So those are just some of the obvious roles that these cells could be having in association with the coral. Corals also associate with fungi. You'll frequently see these fungal hyphae in the skeletons of coral, and there's been some links between nitrogen cycling in the coral holobiome, which is all of the organism and the host combined. But we really don't know a lot about them. And then viruses are another area of this microbiome that can also play an important role in healthy corals in regulating bacterial populations and potentially preventing disease. So these are the total components that we know as of yet of the microbiome. Although I see them, I didn't put the complexions. So I guess I need to change that algae to protists to fully cover that category. But it is quite complex, and I think it's one of the more complex microbial systems that has begun at least to be studied. So my lab focuses on the bacterial and archaeal associations. I was trained in a laboratory that focused on seawater microorganisms, and at the time, and I'm not even going to say the year because I don't want to date myself, but the genomic techniques were really emerging for that system. And so that's what we've been able to capitalize on. Those of us that do study this component of the microbiome, we have some pretty well-developed genomic tools. So our lab's interested in the role that these bacteria and these archaea play in association with healthy corals. So I'm going to start telling you a little bit about the research that our lab is doing in these little secret life stories. And so the first secret life story that I'm going to divulge is that coral microbiomes are highly diverse. So I mentioned forest growers earlier had the suggestion that there were a lot of different bacterial ether types in particular present on corals, but we still didn't really have a real comprehension of what that diversity looked like until recently. And I still think we're not there yet. There's still more to know, but they are highly diverse. The data that I'm going to show you today comes primarily from one technique, and so for the people that aren't familiar with this, I wanted to give you an overview of how we go about even getting this kind of data to evaluate diversity of a microbiome. So we go typically underwater to get a coral sample. We'll bring that back to the lab, extract the genomic DNA. Sometimes we separate mucus and tissue, which are distinct habitats on the coral. We then PCR amplify the small subunit ribosomal RNA gene, which is a really good taxonomic biomarker for much of life, including bacteria and archaea. And then we construct those amplicons or PCR products in some kind of a library for sequencing. Sequencing is done using a variety of approaches. Then there's time spent processing that data. This is the last two steps are generally where most of the time is spent. Quality filtering the data, not all sequences are great. Aligning with this molecule, we have a protein structure that we can actually align it to, and that is very helpful in looking at what are good and error sequences. We spend time clustering and trying to make this data set actually smaller, which is kind of a funny thing that we do, putting species whose sequences are alike together, and looking at the phylogenetic relationships of these microbes, and then producing beautiful graphs of what these relationships look like. So that's a little bit into the workflow that typically goes on with these kinds of data. So I teamed up with a woman, Megan Huggett, a number of years ago to start trying to harness all of the coral microbial sequence data that was being produced, not only by our labs, but also the community. And this really started when I started my lab in Apprill about 8 years ago, not 8 years ago, about 6 years ago. I knew the tools. I had a way to have an insight into everybody's data because they were putting it in these public repositories, and I knew how to integrate it in a way. And I was finding some really cool trends, and I was like, these resources need to be available for other people as well. And Megan agreed, and so we set off to do this project, which basically took us 6 years because the paper just came out yesterday. So hopefully that gives you guys a little bit of an insight into things can often take a while. And especially because we kind of knew the answer to this, so we weren't, we were happy to share it, but it was also, we were taking the knowledge we've learned and already applying that in other ways. So what we did was develop this coral microbiome database. And what we did was just go into GenBank and pull out all the Sanger-based sequences. Anybody know what Sanger sequencing is? It's not totally obsolete yet. From coral-based studies, there were 21,000. I know that doesn't sound like a lot, but what's nice about these sequences is that over 6,000 of them are full length. They're about 14 to 1,500 base pairs. So you actually have a lot of material there for phylogenetic comparison. And this is also a really well-curated database, too, that we're putting together. So we put all these together in this freely available software environment called ARM, which has the secondary structure information of the molecule available as well. So you can assess when you do see changes in the coding structure, if where that is actually happening. So we put all this together, and it's now a resource for our community, which allows for phylogenetic comparison. When you get short reads, you can go into this database and compare them to more established sequences. It's also a really useful tool for primary code design. And this is all laid onto the Silva database background, so we do have information about all the cultivated isolates. Basically, I think it's Silva 119 that this was all constructed in. And then what we did with this database is ask some of the questions that we were interested in too. So one of the questions we were interested in is that who are some of these major players that are falling across many different corals? And Sam, you'll be glad to know this is not just tropical corals. This is deep sea corals, this is octocorals, this is anything that we can call a coral. And we did go back and check and make sure everything was supposed to be added. And so what you're looking at here is a graph. You may not be able to see this, and that's okay, of the number of sequences that fell out with each of these fine-scale taxonomic groups of bacteria and archaea. And the colors correspond to the number of coral species that these sequences were detected over. And the major players in red were the Vibrio, Endozoicomonas, those are both gamma proteobacteria, Rhodoprugaria, a very successful alpha proteobacteria, and then surprisingly, two groups of archaea. We don't hear a lot about archaea with the coral microbiome, but there's quite a bit of data, Sanger-based, because the primers were better, showing that they're there, Thalmarchaeota as well as a Archaeota group. And so this wasn't exactly new knowledge. It's not new knowledge probably to the audience who knows this kind of data now, but back when we constructed this 6 years ago, this was actually new knowledge and really launched my investigations into Endozoicomonas, which I'm going to talk about in a minute. The other surprising thing that came out of this is when we put all this data together across all the known and described phyla of bacteria and archaea, as well as the candidate phyla and groups, we found that the coral microbiome is incredibly diverse. It's home to over 39 of these phylo-like designations. When we started having a look in the literature, and this is just a representation of all the different sequences that they were found in. So you could see like some of the major players, alpha proteobacteria, gamma proteobacteria are showing up, and then we designated between the cultivated isolates and those that were still uncultivated in gray. So when we had a look at the literature, the only thing that rivaled this was sponges. Sponges are actually slightly more diverse. With this kind of a comparison, we found 42 phyla associated with sponges. But when we looked in other kinds of systems, human skin was one of the closest with 19 phyla. Honeybee had about 5. Termites just a handful. And so I think this states two things. One, corals are incredibly, have a breadth of phylogenetic diversity they can associate with. Of course, this is many different species and different kinds of coral. But what it gives me hope to is that there's potentially a lot of functional diversity here too. There's a lot of different ways and different roles that these cells could be interacting with corals. And then the other thing that I will state kind of as a caveat is I think corals are kind of one of the more better studied microbiome systems. And so as we learn more about other systems, it'll be interesting to see how this diversity, how their rise to the top of diversity holds up. And also as we integrate more next generation sequencing data. And that's really the next step of this kind of perspective, taking a more global look at the data that's already been produced. In association with the Marine Biological Lab, we put together this coral microbiome portal. And this is an online resource that I'm going to give a little workshop about this afternoon. This is set up to deal with the next generation sequencing data, primarily, again, still focused on 16-S-based sequences, but it can hold quite a bit more. So we already have some projects in there today, and I'll show you how to look at it. But the nice thing about this is that you don't really have to have a lot of experience with coding or visualization to start looking at these things. And the other nice thing about it is it puts every data set from many different investigators in one framework, so you can easily compare. So I'm hoping with this, we're going to out-compete the sponges and show that corals are even more diverse. So I said you would hear more about Endozoicomonas, and that's my second secret life story today. So this is a story that I teamed up with Chris Bolstra about 6 years ago when we realized that Endozoicomonas were going to be a big thing for corals. And we were able to fund a joint postdoc, Matthew Neve, who spent time in both of our labs. If you don't know Chris, he's a scientist, a very successful scientist at Abdullah University of Science and Technology, and was Monica Medina's postdoc a number of years ago. And so the secret life giveaway that I'm going to tell you today is that Endozoicomonas are very genomically flexible endosymbionts of coral. And I think we're going to be hearing about them for many, many years. But in early looks at coral microbiomes from a disease standpoint, you would have never known that these were associates of coral. But now we are very well established in that. So what did I do with Chris and Matt? Oh, just a little background about Endozoicomonas. So when we started working on them, we looked into the literature, and there was something actually known about them. There were some cultivated isolates from this group. So they fall into the Oceanographic group of gamma proteobacteria. They were first described in 2007 with an isolate from that sea slug. They're heterotrophic, aerobic, use a variety of carbon sources. But there's only a handful of isolates in culture collection. And they're generally associated with marine animals. This is what's very cool about Endozoicomonas. It was named Indo means inside, zoi, and animal, associating with an animal. Sponges, ascidians, corals, and fish, they've only been described, to my knowledge, as pathogens. So here's clusters of Endozoicomonas cells associating with the gill tissues of aquaculture fish, forming these lesions on the surface. So a very interesting group of animal associating bacteria. What we did together is first set off on this global trip kind of around the Pacific and Indian Oceans to look at Endozoicomonas associations with corals. We picked these two corals to study, Stylophora pistillata and Fossopora vericosa, which are pretty abundant throughout these regions. And so we were able to utilize this nice house funding to do a little globe trotting around the world and visit the locations where these two corals reside and take coral samples. When we got them back to the lab, we looked at the 16S RNA gene of these organisms, of the samples from them, and found that about 80% of these colonies did indeed associate with Endozoicomonas. So we guessed right in picking these corals that they were going to have strong Endozoicomonas relationships. What we did with this data, the 16S data, was first took a closer look at the different genotypes of the Endozoicomonas that were in these two species. So what you're looking at here is a heat map of about 57 Endozoicomonas genotypes. These were from minimum entropy decomposition nodes, so this is looking at fine scale differences in that 16S data. So there's a lot of information beyond just that general taxol level if you want to dig deep into it, and MED for us has been a good way to do that. And so what we did was look at these genotypes across these two species across their global distribution. And what we found was some contradicting patterns. So for Fossopora vericosa, in red is a more abundant signature of that particular genotype. We found more similar Endozoicomonas genotypes when we went to all of our different locations. But for Stylophora pistillata, it was actually the opposite. We found regional-based signatures, and some particular genotypes were more abundant in certain locations than others. And these stars are statistically significant distributions between the two species. So you can see there's a whole lot there in Stylophora pistillata. There's a lot of genotypes that aren't present there in Fossopora vericosa. And so we don't know exactly what's driving these two different associations, but we have some ideas, and one could be linked to their reproductive style. So Fossopora vericosa is a spawning coral. It acquires its symbionts, at least bacterial symbionts, from the environment. So it's possible that it's always picking up the same kinds of Endozoicomonas that are in the water. Whereas Stylophora pistillata is a brooder, so it's releasing planula, and these planula compact with symbiodinium. This particular coral hasn't been looked at yet for the transfer of bacterial symbionts, but other brooders can pass their bacterial symbionts. So it's possible that there's more selectivity of the symbionts coming directly from the parents, which could be driving these broader patterns that we see here. So it's just some ideas about what could be going on, but it was very interesting to see these different patterns. We also looked inside the tissues of the corals to try to understand where these cells are residing. So what you're looking at here is an oral section of Stylophora pistillata, so looking down through the mouth. This would be the mouth in the middle. These are the different tentacles. What we're finding are these Endozoicomonas aggregates in the tentacles of these corals. This is done using an Endozoicomonas specific probe that we designed for this project. So if I zoom in on this one, you can see these big aggregates which fluoresce yellow under the microscope. And these are some more individual cells that are kind of broken out from an aggregate. They're residing in close proximity to the nematocysts, so definitely within the tentacles. Corals are kind of a fluorescent nightmare, even if you try to do some corrections. And so we still do histology with some basic H&E stains to also verify what we're seeing is indeed bacterial in origin. So again, similar oral view from Stylophora pistillata, and if you zoom in on this box, you see a similar aggregation of these Endozoicomonas cells. And there's a distinct clearing around that aggregate. So these don't appear to be within the cells of the coral, but they do appear to be within the tissues. We have not done electron microscopy, which would allow us to look closer at that. But very neat that we see these aggregates, and it completely explains some of that high abundance 16S data that we're getting if you have so many cells residing with these corals. We also did the same analysis with Fossilflora varicosa, found the same kind of aggregates. These pictures aren't as pretty. These sections, we identified those aggregates within the gastrothermal tissues. There's some symbiobium cells here, which you can see even better under the with the H&E stain. So this was the first evidence that these are indeed endosymbionts of corals residing in this clear pattern, which is very similar to what we see in the infected fish too, surprisingly. So the last component of this project that we worked on together was trying to get some genomic data on these endozoic.
Would have been good to know.
So what we did was sequence their genomes. We took 3 isolates that were already available in culture repositories. So Alyssa coli is from a sea slug. Montipore is a coral that was isolated in Taiwan. And then, I forget how to say that, Pneumazinsis is a sponge. So we got really nice coverage. This is coverage of the genome, so the size of the genomes for the cultured isolates, but we weren't able to culture this directly at the time from these corals, and so we took a couple different approaches to try to get genomes from them. One was metagenomics, so you're sequencing everything, and this was successful for 2 species, Acropora familis and Stylophora pistillata, Fossilflora varicosa. The genomes aren't as complete in this Fossilflora varicosa. We weren't actually able to separate 2 different genotypes of Endozoicomonas in that one. And then a very expensive but beautiful approach, I think, to get these single genomes is single cell sorting. We did this with the Bigelow lab up in Maine. So we're able to get 2 distinct genotypes of Endozoicomonas from Stylophora pistillata, which evaded all of our other techniques. And so we have, not fully, but a decent amount of the genome, which we think is of these sizes. So what we did was compare all of those genomes as a core genome. This is a pan genome. So looking at what genes are shared between all these groups, there's a lot of gaps. Some of that is field, some of that is due to just the lack of coverage with these techniques. And we found 301 core conserved gene sets. And what we did was compare these core gene sets to what has been known about other Oceanospirales as well as selected symbionts. And so these are significantly distinct enriched gene ontologies that we found for these specific functions. What we found was not exactly surprising, but it was reassuring. So we found a lot of secretion type cells, a lot of transporters. We found a lot of transposase type genes, secretion related genes. And so overall, I think these are difficult to digest individually. So I have this little hypothesis about some insights I have into the functional potential of these cells, because that's really still all we have with these kinds of data. That I think Endozoicomonas are kind of like the taxi cabs of the coral microbiome. So they contain all the genes necessary for all the functions. They're not an obligate symbiont. There in no way can we find evidence of that. So like a taxi, they can carry people to get money. They can have ads. They can be flexible in how they come up with what's needed to survive. They have they're enriched in these genes for transport and secretion processes. So they may be transporting carbohydrates, proteins, amino acids. These are kind of the transporters we found evidence for between the host and other symbionts. Well, there my analogy, I think, works pretty well. They're helping move some of the nutrients and the energy around the coral, just like a taxi does around the city. And lastly, they contain a large number of these transposon genes, or transposase genes. So they may be able to move around elements of their genome to adapt to whatever is needed to survive in that environment. So I think that's exactly what makes Endozoicomonas so successful all these different hosts, as well as other environments, which I'm going to tell you about in just a minute. So maybe this is like branching out into Uber and Lyft and all the things that are needed for the community of new users. So there's still so much, I think, to know about Endozoicomonas, and every time I hear about somebody working on them, I get really excited, because I think that this has been a really successful symbiont. We have to figure out what it's doing now, and then what is it capable of doing next. Okay. So secret life number 3 is a story I'm going to tell you that came from my graduate student, Laura Weber. So she's a student in the MIT Woodsville Oceanographic Joint Program. And Laura got really interested in the microenvironment around corals. And so there's been a couple of studies where have looked at and sampled this, they call it near coral seawater environment, and they found some unique properties different from the reef water. But they primarily use huge volumes of water, like 30 liters, to come up with these observations. And there's just the types of movement on a reef. I don't think you're actually going to capture these kinds of signals if they're really that biologically meaningful. And so Laura really got interested in this, again, using a hypothesis, building on what others have done, and thinking about the seawater surrounding corals. And what she's found is that the seawater surrounding corals may actually help corals recruit their symbionts. So this is my next secret life story. So what did Laura do? Laura capitalized on a cruise opportunity we had in Cuba, and she focused on these 5 Caribbean coral species. And what she did was, each sampling site, she went up to all these corals and sampled within 30 centimeters of the coral. That's within the momentum boundary layer surrounding the coral colony, or what we believe is. She did this with syringes, and she took 60 milliliters of water from the corals. And then we backed off 1 to 2 meters above the reef, still within the benthic boundary layer of the reef, and took water from that location as well. She brought those back to our lab in Woodsville and did the same kind of 16S-based analysis, and then additionally some metagenomic analysis of those samples. So what did we find? What did Laura find? So what this is an analysis, non-metric multidimensional scaling, which is comparing all these 16S-based communities of bacteria and archaea from the coral seawater in pink and the reef seawater in blue. And what she found was that there were distinct signatures. So these circles are the 95% covariance around the mean of the samples within each of those distinct 2 microbiomes. So they were overall significantly different between the two, the coral, near coral seawater microbiome and the benthic reef water, very distinct from Montostria cavernosa. Those don't overlap at all. We didn't have as many samples for Acropora cervicornis, but these are really interesting samples too. I want to study that coral more. It looks microbially very interesting. And within these environments, not only were they significantly different, but when we looked at what kind of bacteria that were living there, we said, yes, okay, this makes sense. Coral seawater had a lot of copiotrophs, bacteria that were good in really high nutrient-rich environments. Endozoicomonas were there, Bermanella, a couple different kinds of gamma proteobacteria, including Alteromonas. So these are just some major signals in the data. Whereas reef seawater had more of the oligotrophic microbes that we find on surface water, SAR11, SAR116, Prochlorococcus, Senecacoccus. Those are phototrophs on reefs, and the NS spore and NS9. So looking much more like surface seawater. So this is really nice evidence from the community standpoint that, again, that corals that do have this really unique microbial community surrounding them. And we're not taking 30 liters of water, this is 60 mils, so it's a much different scale now that we're looking at, which is really neat. Makes me believe those big ones a little bit more, that it's at least similar findings. And then we should look closer at this Endozoicomonas data. And some of these coral seawater samples, this is the percent of relative abundance of the sequences. So some of them had, upmost of 30 to 80% of that microbial community living around the coral as Endozoicomonas. So this isn't mucus we're sampling. These are likely free-living Endozoicomonas that are recruited around primarily Phraites asteroides, but we also detected them associated with all the other species. And then this is the reef water think there was just one sample that had just a little blip. So these are really specifically associated with the corals and just right around those corals. So 7 different genotypes that we detected in the water living there. Some of these do overlap with some of them that we see in Phytes corals. And then what Laura did was, since we had 60 milliliter seawater, we couldn't actually do metagenomics on each of those samples. So we pooled them for species in a defined area. And so what we have are, this is a comparison of enriched or depleted KEGG identifiers for the benthic seawater, as well as the coral seawater associated with all these different species. So we found over 1,000 genes that were either enriched or depleted between these 2 comparisons. And some of the examples of the enriched metabolic pathways in the coral seawater were 2-component secretion systems, bacterial chemotaxis, flagellar assembly, biofilm formation, bacterial secretion center systems, ABC transporters. So, similar things that we were seeing in those symbiont genomes as well. And so this to me really drove home the idea that this is a unique environment for microbes. They're seawater-based microbes. They can live in the seawater, but they're utilizing a coral environment to be successful. Laura is a tremendous artist and illustrator. I'm always calling her into my office for help for things I try to do. And so she put together the synthesis of how she thinks about the microbiome now surrounding corals and all the little sort of hidden processes that we think are happening. So we do think it's a unique environment. We're calling it the coral ecosphere, which suggests that it's enriched for unique microbes and processes. So I'm not sure where I am on time. Feel free to stop me, but I'm going to keep going and finish off with the secret life number 4 story. Microbes play a role in coral superoxide dynamics. This may not be a big newsbreaker to many of you, but I think in thinking about superoxide in coral reefs, it's a new thing for us to think about. This is all work that's been driven by my colleague, Colleen Hansel. She's a very talented biogeochemist who started studying superoxide a number of years ago. And the plots that I'm going to be showing you today, the data products are the result of Julia Diaz, Tong Zhang, who are both postdocs that worked with us, as well as Polina Rab is a new joint program graduate. So Colleen started bugging me as soon as she started her position at Bowie. She actually kicked me out of my office. She was more senior coming in. So I didn't get off on the right foot with Colleen, but she slowly made up for it by saying, Amy, we have to, I gotta work on corals with you. And so we finally sat down and actually designed a project. So a little bit of background if you're not used to thinking about superoxide ROS. So superoxide is indeed a reactive oxygen species. These are oxygen radical intermediates that form during the reduction of oxygen to water. So superoxide, hydrogen peroxide is another radical, as well as hydroxyl radical. Hydrogen peroxide is much easier to measure, it's more stable. The other 2, superoxide and hydroxyl radical, are much less stable. Superoxide has a half-life of seconds. So this is why we don't know a whole lot about this molecule. Superoxide and ROS form both inside and outside of cells. So inside cells, it's a normal component of respiration, but cells can form too much when they get stressed, and some of that too much formation can be accidental. But outside the cells, ROS also forms. Superoxide and hydrogen peroxide are well documented from many bacterial cells. It plays a role in normal physiology. It's a pathogen defense that some bacteria can secrete, and it seems to be play more of a healthy, essential role in these systems. So for corals, superoxide and ROS is primarily thought about with coral bleaching. And so this is just a picture of a reef going from healthy to dying to dead, and the production of superoxide, overproduction within the coral itself, within the symbionts cells, or whatever I'm supposed to call them. I'll get it soon. is leading to this phenomenon. But what what's happening outside corals? We really don't have any evidence until Colleen and I started working together. So measuring coral produced superoxide on reefs, well, way easier said than done. This was our first attempt in Hawaii, and it was actually really successful. But basically, we had to bring this whole instrument, which is laid out here on this little whaler, over to a very shallow coral reef. This is one of the patch reefs in Kaneohe Bay, and put a tube down to the coral. There's a snorkeler holding it next to the coral, and because the half-life is so short, what we're measuring is basically modeling from when it's hitting the tube back to when the sensor is actually detecting it. It's a chemoluminescent-based technique that is measuring this. So it worked. We were like both surprised, but also really excited about how well it worked. This is a typical trace of superoxide. This data is produced in real time on the boat. It's so exciting. Colleen was like screaming while I was underwater trying to stay steady holding this tube. She was so excited. This is a time trace over 850 seconds, and these are superoxide concentrations. This is background seawater. There's naturally produced seawater by the microbes in the seawater. This is a Montipora capritata colony, which is not producing any more that's seen in seawater. And then we have this interface between these two corals. This is Montipora capritata and Phragis compressa. And that's where we start to see a peak. And when you get over to Phragis compressa, there's a very clear signal. You know, a little bit of noise here, but on average, about 40 to 50 nanomole of superoxide consistently produced across that colony. And then we go back to the seawater and it drops back. I mean, we're not like taking out a lot of time here. So this is a really highly sensitive method to measure this. What we did was measure this on 5 different species of Pacific coral. At the time, Kaneohe Bay was undergoing a bleaching event, so I can't really say how healthy these corals were, but we were measuring between bleached and pigmented colonies, and we verified this with symbionts counts back at the lab, dinoflagellate counts. We need a more general term. But what we found was this extracellular superoxide production was not related to bleaching, so we have bleached and pigmented here. These levels are pretty similar, but there's a very clear species-specific pattern. Phragis lobata and Phragis compressa led the pack with the highest amount of production. We went on to do some measurements based in the lab. Colleen took some of the bacteria that we'd isolated from corals. Well, not this one. This was the cultured repository of Endozoicomonas, Rugeria, as well as a Vibrio strain we have, and found that they do indeed produce superoxide. We also looked at coral larvae. So these are annual larvae that do not have symbionts and they do not have bacteria. They're raised in bacterial-free water. So they're also producing abundant superoxide. So we think it's probably a combination of the host and microbes. So because superoxide doesn't cross lipid membranes, most likely the signal that we're getting is from the epithelial cells of the coral and the microbes within that mucus layer of the coral that are combined to produce this. And what we're measuring is a production that's a combination of what's being produced and what is being degraded at the same time too. So there's a lot of superoxide that's mucase and other types of antioxidants in this environment. So it's this whole sort of dynamic, and this is the end result of these kind of dynamics we get. Our most recent development is teaming up with some engineers to develop an underwater superoxide sensor. So we no longer are constrained to shallow corals. Now we have this underwater instrument that we have a little tube now that we just put right up next to the coral, and we can get data underwater. It's still in development. We're in phase 2 of the instrument, but this is some data from our first instrument that we took to Cuba last fall. And so what we found were, again, really species-specific signals. So this is Phragis asteroides and Phragis, which are, again, making a lot of superoxide. This is, like, insane, like 160-ish nanomoles of superoxide. That's never been measured anywhere, I think, for an organism. So they're producing a lot of superoxide. And then there's various amounts of production by these other Pacific corals. Some are actually showing negative because it's less than what's in the seawater surrounding them. So we think that there's more degradation processes going on there. So what's neat about this is now we have coral systems that obviously make a lot and don't make a lot. And so it provides us an experimental system to start to understand why they're making a lot, who's making it, and start looking at some of those questions. But this is, I think, such an exciting area, and now I'm starting to think about corals as having sort of this, especially Crites, this zone of superoxide surrounding them too, which could be playing a big role in why they're successful and they're able to evade bacterial colonization. So lastly, I just want to leave you with a summary of these. Hopefully now it's a little bit of a less secret life of coral microbiomes. Coral microbiomes are indeed highly diverse. We're still working on that number, but it's getting up there. Endozoicomonas are a major portion of the microbiome of many tropical corals. They're also a really genomically flexible endosymbiont. And I think we're just at the beginning of understanding what their role really is. So corals. Seawater surrounding corals may be helping them recruit symbionts, maybe even pathogens. We're not sure about that, but I think there's a lot of research now that can be done with this interface between the coral and the environment. And microbes, bacteria associated with corals can play a role in the superoxide dynamics within corals, and we have a lot of research yet to understand really what's going on there. So I try not to show dying corals in my briefs and my talks because I get sad, but I did want to make a point that the reefs are not doing well today, right? We've lost over 50% of corals on reefs due to bleaching, due to disease. A lot of this has happened in the last few years. This is an ecosystem under incredible threat right now. And I think we're just beginning to understand like what these microbiomes look like. And I just showed you a very small component of that total microbiome pie to corals. And I get pretty nervous when we're talking about solutions using the microbiome to try to help coral restoration and help corals become more successful. I think that's a great goal, but there's so much still that we need to know about these. So we need more people. I love the graduate students that are working on these systems and related systems. And we obviously need more money and policymakers to help change the research avenue for reefs. But overall, it's an exciting area of research to be in. And I just lastly like to thank the people from my laboratory. I tried to acknowledge all the individual contributors to the results, but we also had some contributions from some technicians in the lab, as well as Rita Rachmawati, produced our data for those Indonesian coral samples that we looked at, and my collaborators and the funding sources. Thank you all for your attention today.
I was quite impressed with the aggregates of the
Endozoicomonas. Yeah.
Compliment. So I was curious whether you looked into what the transporters could potentially use in substrates for bringing in nutrients, because that might give you insight into what the host is providing in those areas. Maybe you can grow those in cultures even better.
Yeah, absolutely. So one of the transporters that I'm really interested in appears to be able to transport cellulose. So algal cell degraded products, right? And so potentially, I'm not really sure that this aggregates in the tentacles, there's not some biodinium there, but potentially they're there because the coral is eating, there's some sloppiness going on, and maybe they're able to manage that. But yeah, I agree. That's we'd love to connect the genomics back to that. Question?
So on that same subject, are those aggregations of the bacteria, are they in a pocket in the endoderm? Or are they interested?
It appears to be in a pocket in the endoderm, but again, we haven't done the electron.
So kind of like the pockets of Vibrio in squid.
Yeah.
I didn't see it there. So on another related note, your comparison between Stylophora and Positophora, I think I might have some insight that would be easy to track down.
Cool.
So I know that Viricosa and Positophora viricosa is a very widespread species from the eastern Pacific all the way to the western Indian Ocean. Whereas Stylophora pistillata, which species were you doing? Because there's probably 6 different species of Stylophora pistillata that with a simple sequence, and they're regionally located. So you have the Red Sea, and you have Pacific and Indian Ocean style off. So it would be really quick to say, okay, well, you're dealing with a different species of Stylophora, and that's what gave you that really diagnostic pattern in those regional diagnostic pattern. So that would be simple.
Yeah, that's a great idea. We did do some host genotyping, but I don't exactly remember the marker, and I understand the level that we were doing at
There's a paper I can
okay, cool.
look at.
Yeah.
It's really
Yeah, I mean, I know at least the one in American Samoa.
yeah. Oh, they are.
Yeah.
That's it would be easy to do.
Yeah. Cool. Yes,
I don't know what to start asking first, but so the superoxide concentration of praise. So praise, well known to be best being what's often described as. So, did I understand right that what you're suggesting is that already has high tolerance to superoxide concentrations, getting so. I mean, that's one of the ideas we have, right? Is reaching this part of the cause. Concentrations in how by differentiation. So is that your thought? Is that the host is already able to feel it?
yeah, potentially. I mean, these are two very different pools of ROS. What happens inside the cell doesn't leak out outside. What we're measuring is what's coming off. So I think, at least on the surface, they do have this unbelievably high concentration. Whereas corals doesn't inhibit their growth. There doesn't appear to. But I don't think we can link it back to what's happening inside the cell. Yeah. And, I think potentially it does have some kind of a role, some kind of resistance, right? Whether it's bleaching or other kinds of phenomena. But those two pools, we can't.
You tried measuring the iron. Studies of iron.
Yeah, so around these corals, we did collect water, and it would cone stuff like that. So it wouldn't vary a lot between these cones in all the same kind of environments. But yes, it definitely does. And Colleen's done really nice work studying like different seawater environments. She thinks the iron and mercury is another one that plays a role in some of the dynamics there. Sure, yeah. Yeah. You think that would vary a lot with corals?
Oh, yeah, it does very well with corals. I'm really interested in contact and like for both of us, iron.
Okay.
Yeah, varieties is. I'll take a look at
Oh.
it before we chat.
Yeah, that's good. These are things you don't think about a lot. How much these guys might vary on certain. Yes.
I was wondering if you, with the peroxide, have you looked at the same species in like different groups? See if it's like, a genetic like institutive production of this, or is this like particular conditions that species seem in that reef that causes these higher levels?
So Prides asteroides we have the most data on across really different regions. And so we have the field data from Cuba, which is across quite a distance of reefs. Those are all pretty consistently high. I mean, there are some variations like 20 animal. And we also have some data from Prides asteroides, a very different location. Those are taken in a lab out of an environment and also have really high. So I think the environment has some subtle effects. So, maybe there's some signals that we see are really consistent, at least with primes.
Do you think of a way to control for surface area of coral like the kind of flatter corals, lower peroxide surface areas. Maybe that's to do with just how much coral surface area is contact with the water. The peroxide is coming from.
Yeah, so we haven't figured out a good way to do that yet. I mean, we're basically holding a tube over the surface, so we're getting a sort of integrated signal. We tend to work on like an area that's not pretty homogeneous, like we're not taking like a tip of the word. So I think what we're doing is sampling a pretty similar area, but there's so much variation in like polyp size and depth and all of that as
Like,
well.
like between two branches of variety, kind of getting superoxide. Like next to Montana, you really only near one.
Exactly. It's pretty, so the laboratory studies we did, like we pulled the tube away about 30 to 40 centimeters away. You see the backgrounds. You have to be almost right. We've occasionally there's so many fun things you can do now that it's underwater instrument, right? And the instrument works okay, but it was starting to like heat up after about 20 minutes. 20 minutes of data. It's still under development.
So besides PhD, have these corals that and I don't know it.
Yeah, they
There was
do. We haven't done that. I mean, we'd love to do that. I think that like some packaging exposure experiments, all that would be dynamics. So as well as antioxidant use.
right.
Yeah, they're pretty straight. They don't get as many diseases, and that's exactly the trends that we're seeing sort of match on the larger health.
So I'm interested in near coral environments, and I don't know much about ocean environments, but I'm wondering if you have thought about or controlled for bulk flow past the flow. Like shallow ones might have more bulk flow.
Yeah, no, that's a great point. We would love a better connection between the flow environments. There's been like one really nice hydrography study over Apprill. These are like huge friends. Corals. So I think it's quite difficult to get a really nice flow regime mapped out. And we had some early hopes of doing some like experiments and plumes, looking at particles and how they might move over coral skeletons. And it could be pretty challenging to think about. But I think at least like benthic flow and thinking about like really different flow regimes, like a flow regime battery, that would most of these environments. Deeper forms.
Thank you all for coming and